how are you validating your processes to ensure consistent removal of residual impurities across different batches of allo cell therapies? Bruno, thoughts on that?
Right. And again, just starting with the foundation that Pratik just went through is a big part of the quote unquote validation exercise, albeit theoretical. And then beyond that is, again, something that we already touched on here, is collecting as much data as possible as you go through the scale up path, right? So, I mean, as early as research scale experiments, start collecting as much data as possible. And then as you move into, let's say a process development lab or your manufacturing demonstration batches that will produce material for IND enabling toxicity studies, we definitely collect those. And then as we move into a GMP facility, right, usually we start with some engineering batches and then eventually we begin GMP manufacturing. And it's around that time that we compile the data for regulatory file submission. And so if you follow that paradigm of data collection, you'll have a rich data set that you can use to verify and validate this theoretical frame explained, and it's usually very well received by regulators.
And I will actually comment there. You brought up a really excellent point. I think that one of the things that most cell therapy companies have learned, and certainly is true, is generate all of that data and go to the agency for these discussions with data in hand and state what you would like to do or what you believe is sufficient for your product. I think that what none of us want is to go to the agency and say, what do you think we should do? Because then you may get an answer that is either not achievable or doesn't align with what your practice is. So I think going in with data and saying, this is what's feasible we can look for, this is what we can't look for, this is why we believe that it's safe, this is why we believe that it's at an acceptable level, and showing all that data, I think the agency has become very sophisticated in their cell therapy discussions. And I think that is something that we've seen is when you go with data and you really discuss your plan and the logic behind it, we find that they're very receptive and will work with you, because they do want to advance these therapies as much as we as companies want to advance these therapies to patients. So, get your data early, get it often, and then go prepared.
Wonderful.
I think both Bruno and Kate have answered the question really well, but maybe one thing I'll add is around the data itself. Oftentimes what people will do is, let's imagine your process calls for a single wash step. And so you have you're determining concentration for a particular component before and after. It behooves you as a developer of that therapy to maybe go beyond that one wash cycle, look at a second or a third, and see what kind of decrease do you see in the residual concentrations. Not because you want to change the process, but you have an understanding of what exactly are you able to achieve with that unit operation. That is the kind of information I think that both Kate and Bruno are pointing to. That's data-rich information that can help really determine what's the most appropriate way. If it turns out to be that early on you have a dose determined, but over time you're going to change that dose, well, then maybe you have to redo the calculations, and certainly you do, and then maybe you need that extra wash step. If you had that data set already from the beginning, you know what that can achieve. And so you're in a much better spot to be able to make adjustments if you need to.